Hi, in this video we will perform the analysis regarding estimation of genetic parameters like genotypic, phenotypic, coefficient of variance, heritability, genetic advance, and other things which are very much important for estimation of these genetic parameters like phenotypic genotypic correlation and phenotypic genotypic path coefficient analysis. Well, this kind of analysis is done usually by the people who are engaged with plant breeding trials, and in case of forestry, people who are engaged with forest biology and tree improvement works. So in order to perform this analysis, I'll be using a data set. For that I have to import that data set which is available on my desktop. Then I'll click where it is. This is my data set, and I will click on this open. Once it will get displayed here, I will click on this import option. Then okay, the data is here. Then I will click on this import option. Here we have the data. So we have genotypes. We are having 10 number of genotypes, 4 number of applications, and we have variables like seedling height, collar diameter, internodal length, leaf area per plant, then root length and number of branches per plant. So we want to evaluate all these 10 genotypes utilizing all these variables. For that we'll be using a library which is known as library variability, a very wonderful library for estimation of genetic parameters. For that first we will write names. Geno, this is the name of my data set. So we have 8 variables. The first 2 are genotype and replicates, since we'll be performing the analysis from 3rd to 8th. The first 2 are genotypes and replications, and the position of the variable is very important in this kind of analysis. For that, in order to estimate the genetic parameters, we will use the default code here, gen.var, followed by the name of the data. The name of the data is gen, and I will put these square brackets here. This is geno, then square bracket, and within these square brackets I will write 3:8. The reason is that since my variables start from the 3rd column up to 8th column, that is why 3,8, followed by again the name of my data set. I'll put the $sign and click on this genotype. Then again I have to use another vector again here, since there are 2 vectors here, one is genotype and another one is replication. I will click on this enter, and we have the results for all the variables. So we have say for example for seedling height, we have an OR table. Here you can see the genotypes are significant at 5% level of significance, and we have the results for genetic parameters like grand mean, standard error, and especially these are very important like environmental variance, genotypic variance, phenotypic variance, from which heritability variables are values are calculated, since heritability is the ratio of these two. And we have the results for values for genotypic and phenotypic variance, as well as the heritability in the broad sense, not in the narrow sense, but we have results for broad sense heritability. We have results for genetic advance. We have results for genetic advance as percentage. So within one click you will get the result for all the variables. So this library is very important. So apart from this, we can perform the analysis like if we are interested to perform the phenotypic correlation, since in estimation of genetic parameters we are also interested to see the relationship between the variables genotypically and phenotypically also. For that again we will write the name of this data set. Then we'll be using the same step, 3:8, like we did in the previous step for calculation of estimation of genetic parameters. Then again the name of the data set, then genotypes, followed by the name of data set. I'll put the $sign and the rep. Then I will click on this enter. So we have the results in terms of phenotypic correlation for these all variables. If we are interested to perform the genotypic correlation, for that what we have to do, geno.corr. Okay, then again we will click on this. we'll get the results for genotypic. And if we are interested to perform the phenotypic path coefficient and genotypic path coefficient, for that we have to decide what is the position of our response variable. Say for example, I will take the last one, the 8th column, as my response, the study variable, variable of interest, and rest 3rd, 4th, 5th, 6th and 7th as auxiliary or explanatory variable. So in order to perform that, I will write pheno.path, then I will again write the name of the data set, but within the square brackets I have to first write the position of my response variable, then followed by the name of this geno, and within these square brackets I will write 3:7 now, since the 8th variable is my response. Then followed by the same thing, geno, we have to create a vector of this genotype, then the vector of these replications. Then I will click on this. We have the results for phenotypic path coefficient. The diagonals are the Dirac effect, and the indirect effects are above and below the diagonals, and we have the values for residuals also. Then if we are interested to perform this genotypic path, geno.path, then again we'll click on this, then enter. So again, in the similar way, we have the results for direct and indirect effects. So this kind of a package is very helpful for those people who are interested to estimate the genetic parameters like genotypic, phenotypic, coefficient of variance, heritability, genetic advance, phenotypic, genotypic correlation in a very quicker and short time. I hope this video will be benefited. Might find benefit from this video. Thank you.